I want you to imagine taking a perfectly healthy marathon runner. You change absolutely nothing about their muscle mass, nothing about their lung capacity, and you leave their cardiovascular system in just pristine condition.
Right. I mean, that sounds like someone ready to set a world record.
Exactly. But what if you simply turn off one specific microscopic part of their immune system? According to this really groundbreaking 2026 paper we are diving into today, that runner's physical endurance wouldn't just dip slightly.
No, not at all.
It would drop by 50%. I mean, they would literally collapse halfway through the race.
Yeah, and it is a complete biological paradigm shift. I mean, for decades, we've treated sports science and physiology like a straightforward engineering equation.
Like checking a car before a road trip.
Exactly. If you want to know how far a car can drive, you check the engine, which is the muscles in this case, and you check the fuel pump, so the heart and the lungs. And if those are in good shape, you have your endurance.
Right.
But this paper, which was published in the journal Cell, it proves that equation is missing a massive, unexpected variable.
And that variable is the B cell. The white blood cells whose famous textbook day job is just sitting around making antibodies to fight off viruses and bacteria. So the mission of this deep dive is to unpack this totally newly discovered, completely immune-independent role of B cells.
Yeah, it's wild.
We are going to explore how they secretly regulate your exercise capacity through this incredibly complex, high-speed chemical conversation between your immune system, your liver, and your muscles.
And the implications here go way beyond just athletic performance. I mean, in human medicine right now, B-cell depletion therapies are incredibly common and, well, they're highly successful.
Right, for things like autoimmune diseases.
Exactly. We use monoclonal antibodies, specifically anti-CD20 therapies, to purposefully wipe out B cells.
Yeah.
And it treats severe autoimmune diseases like multiple sclerosis and systemic lupus, as well as certain lymphomas.
But this deep dive asks a completely fresh question, which is, when we wipe out those B cells, what happens to the rest of the body's physical performance?
Right. What's the collateral damage, essentially?
Exactly. So we're going to take you on a step-by-step journey through this biological mystery. And we'll be doing mini recaps, bringing in some analogies, and even tossing out questions to test your understanding along the way. So let's start with the initial discovery. It really reads like a medical mystery.
It really does. So the researchers wanted to see what happens when an organism loses its B cells. So they set up an experiment using just, perfectly healthy mice.
And they had to be really thorough here, right?
Very. To ensure their findings were airtight, they removed the B cells using two distinct methods. First, they used a genetic model known as MUM-MT mice.
Okay, MUM-MT.
Yeah, these are mice that are genetically engineered from birth to basically lack mature B cells. But genetic models can sometimes have unseen compensatory mechanisms, right? The body adapts. So they also used a clinical method.
To make sure they weren't just seeing a genetic quirk.
Exactly. They took normal, wild-type mice and injected them with anti-CD20 antibodies, which, like we mentioned, directly mirrors the therapies used in human hospitals to wipe out the B-cell population.
Okay, so we have these mice, no B cells, but otherwise they are standard, totally healthy animals. The researchers put them on treadmills and rotorods, which, if you don't know, are essentially these spinning cylinders designed to test a mouse's balance and stamina.
Like a log rolling competition for mice?
Yeah, exactly. And the results are completely shocking. These B-cell depleted mice just fail. Their exercise capacity vanishes by like 40 to 50%. Even their basic grip strength just plummets.
It was a profound and immediate decline. I mean, they simply couldn't keep running.
Okay, let's unpack this because I want to make sure I'm picturing this right. We take away their immune cells and their muscles just give up.
Yeah, that's what it looks like.
But my immediate gut instinct is that, well, if you remove a massive chunk of the immune system, maybe the mice just got sick. Yeah. Like if they're fighting off some underlying infection, of course they're going to be exhausted. Did their muscles atrophy from illness? Did their hearts struggle to pump?
It is the most logical assumption to make. But the researchers anticipated that, and they ran a full diagnostic workup. And the mice weren't sick.
Not at all.
Not at all. Their lungs were totally clear. Their muscle size and the actual morphology of their muscle fibers were completely indistinguishable from the healthy control mice.
Wow.
And their hearts were pristine. I mean, the researchers measured the ejection fraction, which is essentially the metric of how powerfully the heart pumps blood out into the body with each beat, and it was totally normal.
So the pump is working perfectly.
Perfectly. They even looked at the capillary density, the network of tiny blood vessels delivering oxygen to the muscles, and they found zero deterioration. The T-cell levels were fine. Everything was fine.
So we have a healthy engine. We have a healthy fuel pump. The structural integrity of the car is perfect, but the car just stops running.
It's a pure, isolated failure of endurance.
Okay, so time for a quick 5-minute mini recap. Here are the bullets. B cells are removed. Physical performance drops by half, but heart and muscle structure remain perfectly fine. So here's a question for you, the listener. If the engine, the muscle, and the fuel pump, the heart, are perfectly fine, what is missing?
Right. If it's structurally sound, the missing link has to be something invisible.
Exactly. Which brings us to section 2.
Right. This forced the researchers to become metabolic detectives. They utilized a highly advanced technique called liquid chromatography mass spectrometry, or LCMS for short.
Let's break that down, because it sounds a bit intimidating. As I understand it, it's essentially a way to take a hyper-detailed chemical inventory of the body, right?
That is a really great way to describe it, yeah. Whenever your body breaks down food or builds muscle or, generates energy, it creates tiny molecules called metabolites. So LCMS, it separates all the complex liquids in a tissue sample, that's the liquid chromatography part, and then it weighs the individual molecules using a mass spectrometer.
So it's basically weighing the chemical fingerprints.
Precisely. It allows scientists to map out thousands of metabolites at once. They looked at the skeletal muscle and the blood serum of both the healthy mice and the exhausted B-cell depleted mice, basically searching for an anomaly. They found 5 distinct metabolite modules in the muscle and 4 in the serum.
It's kind of like looking at 2 identical photos and trying to spot what was photoshopped out. If you're looking for a missing suspect, you look for what disappeared.
Yes.
And they found a glaring omission. In the B-cell depleted mice, one specific amino acid had absolutely plummeted in both the blood and the muscle.
Glutamate.
Right, glutamate or glue. But discovering that glutamate was missing was only half the puzzle, because while glutamate was dropping in the muscle, its chemical precursor, a raw amino acid called glutamine, was piling up in massive quantities in the liver, like cars in a traffic jam. So if glutamine is piling up in the liver, I'm guessing the conversion process just stopped.
Exactly.
To prove that, didn't they track the actual molecules moving through the body?
They did, using a really elegant technique called heavy isotope tracing.
Oh, this is so cool.
It really is. They took a form of glutamine where the standard carbon atoms were replaced with carbon-13, which is a heavier, stable isotope. We call it U13C-GLN. Because it's literally heavier, that mass spectrometer we mentioned earlier can easily distinguish it from the body's natural carbon.
It's physically heavier, so it stands out on the scale.
Right, so they infused this heavy glutamine into the mice and simply watched where the heavy atoms traveled in real time.
I love that. It's like attaching a microscopic GPS tracking device to the raw materials. And what did the tracking data show?
It proved definitively that the skeletal muscle was entirely blameless. The muscle's metabolic machinery was perfectly capable of burning fuel if it had it. The problem was entirely upstream.
The liver.
Yes. The liver had fundamentally stopped doing its job.
Mm-hmm.
It was no longer taking the raw glutamine from the blood and converting it into the glutamate the muscles desperately needed.
So a mnemonic visual for you to remember this. Imagine the liver is a massive factory. Glutamine, with an N, that raw amino acid, is the unrefined material being dropped off at the loading dock. Inside the factory, workers process it into glutamate with a T, which is the high-octane or rocket fuel.
That's a great analogy.
Right, and that fuel is then loaded onto trucks, which is the bloodstream, and shipped out to the muscles down the road. But when you wipe out the B cells, the factory manager basically goes on strike.
Yeah.
The unrefined glutamine piles up at the liver's loading dock, the trucks run empty, and the muscles just completely run out of gas.
That captures the dynamic perfectly. And the researchers proved this supply chain theory with a remarkably satisfying aha moment, a rescue experiment.
What did they do?
They took these exhausted B-cell depleted mice, the ones who were failing on the treadmills, and they bypassed the liver factory entirely. They just fed the mice a diet highly enriched with readily available glutamate.
So just putting the fuel straight into the system, bypassing the strike, and their performance bounced back.
Completely. Their exercise capacity returned to baseline. They ran just as long as the completely healthy control mice. their grip strength returned.
Wow.
It was a total metabolic rescue, proving that the fatigue wasn't an irreversible structural damage. It was just a severe fuel shortage.
Okay, mini recap time. Number 2. B cell loss causes a glutamate shortage because the liver stops converting glutamine into glutamate. Feeding the mice glutamate fixes the fatigue. So question for you. How does a B cell, which is literally just a white blood cell floating around in the bloodstream, how does it talk to a giant organ like the liver to keep this factory running?
And that leads us to the messenger and the machinery.
Exactly. Section 3. We have to find the chemical blueprint. How does the B cell communicate? And the answer is a signaling molecule called TGF-beta1. That stands for transforming growth factor beta-1. B cells secrete this directly into the blood.
Right. And what's fascinating here is the exact molecular machinery, because they left no detail behind. When TGF-beta1 arrives at the liver, it acts like a boss handing out a very specific genetic transcription order.
Okay, what's the order?
It forces the liver cells to heavily upregulate 2 specific pieces of machinery. First, it activates a gene called GLS2, which produces an enzyme known as glutaminase 2.
Okay, so I think of GLS2 as the molecular scissors. Its sole job is to physically chop the raw glutamine into that high-octane glutamate.
Exactly. But making the glutamate isn't enough, right? You have to get it out of the liver and into the blood. That's where the second upregulated gene comes in. SLC7A5.
SLC7A5. Okay, this is a shipping door, right? An antiport transporter.
It is.
The best way to visualize an antiport transporter is like a molecular revolving door embedded in the wall of the liver cell. It's an exchange mechanism. So as it pulls one amino acid into the liver from the blood, the revolving door spins, physically pushing the newly manufactured glutamate out into the bloodstream so it can rush to the muscles.
Perfect description. And the researchers didn't just guess this was the pathway. They dismantled it piece by piece to prove it.
How so?
Through some truly stunning genetic engineering. They created mice where they knocked out the ability to make TGF-beta1 only in the B cells. The rest of the mouse was totally normal. Then, in a separate line of mice, they left the B cells alone, but knocked out GLS2 and SLC7A5 only in the liver.
So in one group, they silenced the text message. In the other group, they broke the molecular scissors and the revolving door.
Exactly. And in both of those highly specific, surgically targeted scenarios, the mice exhibited the exact same profound exercise failure as the mice who had no B cells at all.
It's a perfect chain of command.
It confirmed it flawlessly.
Yeah. Oh, wait, let me push back on this for a second, though. Anyone who studies exercise physiology knows that intense exercise naturally causes TGF-beta1 levels to rise in a normal body anyway. Muscle damage and other tissues release it when you work out. Are we absolutely sure B cells are the main source orchestrating this during a run? Couldn't the muscles just be signaling the liver themselves?
The researchers asked that exact same question. Yes, exercise naturally causes a spike in TGF-beta1. But here is the definitive proof. In the mice lacking B cells, that normal exercise-induced spike just never happened.
Oh, really?
It was completely suppressed. This reveals that B cells aren't just casually contributing to the signaling pool. They are the master regulators here. They override other tissues to ensure this specific metabolic pathway activates during physical exertion.
That is wild. Okay, mini recap 3. B cells secrete TGF-beta1. The liver receives it. The liver makes GLS2 the scissors to create glutamate and SLC7A5 the revolving door to export it. So question for you. Once this glutamate fuel finally arrives at the muscle, how exactly does it make the muscle run longer? Which brings us to the final destination, section 4, the muscle engine. So explain what happens when the glutamate actually arrives at the skeletal muscle. Does it just burn it like coal in a furnace?
No, it is so much more sophisticated than just burning calories. When glutamate arrives at the skeletal muscle, it doesn't primarily act as a caloric energy source. It acts as an intracellular signaling trigger.
A trigger for what?
In the muscle, glutamate fosters something called calcium oscillations, or calcium sparks.
Let's expand on that, because calcium is the absolute key to how humans move. You know, when you want to flex your bicep or take a step, your brain sends an electrical impulse to the muscle. But that electricity has to be translated into physical movement.
Right.
It does that by triggering a flood of calcium ions inside the muscle fiber, which physically bind to proteins and force them to slide past each other, creating a contraction.
Precisely. You cannot move without those calcium sparks. And the glutamate arriving from the liver dramatically enhances and sustains these calcium oscillations. And here's where we see the domino effect, the climax of this incredible biological cascade.
Okay, lay out the dominoes for us.
Right, so domino 1, glutamate triggers the calcium sparks. Domino 2, Those enhanced calcium sparks activate a specific protein kinase known as CAMK, which is calmodulin-dependent protein kinase.
KMK. Let's call KMK the master contractor of the muscle's power grid. So when the calcium sparks wake up the contractor, what exactly is it hired to build?
Domino 3. It is hired to build and maintain the mitochondria, the microscopic powerhouses inside your cells. When CAMK signaling is active, it drives mitochondrial biogenesis. It's literally forcing the cell to create new mitochondria.
Wow.
But just as importantly, it maintains their internal architecture. Specifically, it keeps the cristae sharp and functional.
The cristae, those are the tightly packed maze-like folds inside the mitochondria, right?
Yes. The folds exist to maximize the internal surface area. The more surface area you have, the more biochemical reactions can take place to produce ATP.
Which is domino 4.
Exactly. ATP adenosine triphosphate is the actual fundamental unit of cellular energy that your muscles use to execute those contractions. Healthy mitochondria produce abundant ATP. Maximum ATP means you can run for miles without stopping.
Which really brings us back to the tragic contrast in our B-cell depleted mice. Let's look at the catastrophic domino effect of what happens when this system breaks down. Without B cells, there's no TGF-beta1 text message to the liver. The liver factory shuts down and no glutamate is shipped to the muscle.
Right. And without the arrival of that glutamate, the calcium signaling inside the muscle drops. It becomes weak and erratic.
So KMK, the master contractor, goes quiet. It essentially falls asleep on the job.
Yeah, the power grid stops getting maintenance. It physically deteriorates. When the researchers looked at the muscles of the B-cell depleted mice under a high-powered electron microscope, the imagery was stark.
What did it look like?
The mitochondria had become blurry and swollen. Those beautiful, sharp cristae folds were damaged and disintegrating. Because the internal architecture was crumbling, the mitochondria couldn't produce enough ATP. ATP levels plummeted and the mice simply ran out of gas.
And just to bring it full circle, when they fed this failing mice the glutamate diet, the electron microscope showed those blurry, damaged mitochondria completely heal and rebuild their cristae. It's an absolutely flawless biological baton pass.
It really is.
Okay, final mini recap. Glutamate hits the muscle, spikes the calcium, activates KMK, builds healthy mitochondria, makes ATP, and powers exercise. So question for you, can you see the incredible relay race happening inside you right now? B-cell to liver, liver to muscle, muscle to mitochondria.
It highlights a level of biological integration that we're only just beginning to map.
So what does this all mean? Let's bring this complex mechanism back to your daily life. This isn't just a mouse study. This introduces a massive concept into human physiology called immunoexercise. The idea that your immune system and your physical fitness are intrinsically metabolically linked.
And that has immense, immediate clinical implications. Think back to those human therapies we discussed in the very beginning of the deep dive. Right now, there are patients worldwide receiving anti-CD20 therapies for lupus, multiple sclerosis, or lymphoma. We are actively depleting their B cells to save their lives.
And if you talk to these patients, one of the most massive universal complaints is profound, debilitating physical fatigue.
Historically, medicine has often chalked that fatigue up to just the general malaise of fighting a chronic illness.
Right, like, of course you're tired, you're sick.
Exactly. Or they think it's a vague side effect of harsh medications. But this research provides a glaring spotlight on a potential reason for their exhaustion that is purely metabolic. It might not be vague malaise. Their liver factory might literally be shut down, starving their muscles of glutamate.
Which is incredibly hopeful news, because if the fatigue is driven by this specific metabolic bottleneck, it suggests a tangible solution. It suggests that something as surprisingly simple as dietary glutamate supplementation or targeting this specific liver pathway could actually help these patients regain their strength.
It's the best kind of science.
Yeah.
It answers a fundamental biological mystery while simultaneously opening the door to improve patient care.
Absolutely. So we want to leave you with a final provocative thought to mull over. We love to map the human body like a globe, dividing it up into neatly labeled isolated continents. The cardiovascular system is here. The immune system is quarantined in the lymph nodes. But if B cells, the famous antibody producers of the immune system, are secretly moonlighting as the managers of your liver's metabolism and your muscle's endurance, what other traditional boundaries in human biology are completely wrong?
Yeah, what else are we missing?
What other organs are secretly talking to each other behind our backs? Next time you go for a run, maybe don't just thank your lungs and your legs, thank your immune system.